Good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Translational Research in Oncology, Innovations in Model Development. My name is Aisha Rashid, and I will be your X-Talks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. We also have a handout available for this presentation, which you can download through the Handouts tab in your control panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Certis Oncology, who developed the content for this presentation. Certis Oncology Solutions is a life science technology company committed to realizing the promise of precision oncology. The company's product is oncology intelligence, highly predictive therapeutic response data derived from advanced biological models of cancer. The company's proprietary platform informs individual treatment decisions and accelerates the development of new cancer therapies. The company partners with therapeutics developers to help close the problematic translation gap between preclinical studies and clinical trials. Certis brings greater certainty to go, no-go development decisions through more clinically relevant, well-characterized, and annotated models combined with advanced imaging technology. And now it's a pleasure for me to introduce our speakers for today's event. Dr. Nakashima is an accomplished cancer researcher and recognized pioneer in orthotopic cancer model development. At Certis, he provides thought leadership, scientific guidance, and supports the company's ongoing R&D initiatives. Dr. Nakashima has published several scientific posters and papers in cancer research, most notably on the clinical utility of orthotopic patient-derived xenograft models. Under a CIRM fellowship, he received his doctorate in molecular and medical pharmacology at UCLA. Dr. Nakashima completed his postdoctoral training in the Department of Neurobiology, where he received a Kirstein National Research Service Award to study cell interactions in the brain-tumor microenvironment. Dr. Sperry is a translational research scientist with deep experience innovating models of disease, including inflammation of the CNS, which contributes to neurodevelopmental disorders, neuro-oncology, and preclinical animal models. At Certis, he is responsible for advancing the clinical relevance of in vivo, ex vivo, and in vitro cancer models, as well as the design and execution of translational studies for drug development and personalized medicine. Dr. Sperry has published several impactful scientific papers in cancer research, most notably on modeling glioblastoma. Dr. Sperry received his doctorate in molecular and medical pharmacology at UCLA. Dr. Srimali is an accomplished immunologist and cancer immune therapist who brings over 20 years of expertise in the field of immuno-oncology. At Certis, he develops and validates biomarker-based PDX models across different immune therapeutics, including CAR-T, NK immune checkpoint inhibitors, and bispecifics. Dr. Srimali has held scientific and leadership positions at NCI-NIH, MD Anderson, and various pharmaceutical companies. His research has been widely recognized in peer-reviewed journals, patents, clinical trials, FDA approvals, Federal Transfer Technology Awards, and scientific speaking engagements. He received a PhD in biochemistry from ICMR, Mumbai University, India. And now, without further ado, I'd like to pass over the mic and the presentation to our speakers. You may begin when ready. Dr. Nakashima, I believe you're on mute.
Hi, can everyone hear me okay?
Yes.
Aisha,
Yes,
we just had a little technical hiccup right here, so we're gonna have to change things a little bit on the fly, but we're good to go. So,
okay.
thank you everyone for joining us this afternoon. Today we're going to discuss how custom models can help improve the success rate of developing cancer therapeutics that ultimately get FDA approval. We're going to start first by overviewing some of the challenges that exist in drug development and discuss opportunities that exist to improve the outcome, primarily using custom preclinical models. Now, there are a number of different options within the preclinical toolkit, so to speak, and we're just going to highlight a few of them. We're going to review some specific case examples here that we've performed here at Certis, and talk a little bit more about our company, and then finish with a Q&A. So one of the major challenges with developing cancer drugs is that cancer is not one disease. It's many diseases, hundreds. This is well known throughout the cancer community now, that heterogeneity within patients, heterogeneity within the tumor itself, heterogeneity within the genetics of a tumor, poses a challenge when developing cancer drugs, especially when these drugs are kind of made for one size fits all. This has led to, or one of the major reasons why there is such a dismal success rate for FDA approval of less than 4% of all cancer therapeutics. Of nearly the 8,000 compounds that are currently in development, it's estimated that at this rate that less than 300 will get an approval. And as we learn more about cancer and more how complex it is, how complex cancer is, the mechanism of action of these drugs therefore also need to keep up with the complexity of the biology. And it's also one of the other challenges that is emerging is that the field of biomarkers to help predict what cancer type will respond to a certain drug still remains elusive, meaning that a proposed biomarker for a patient, once that patient goes on that therapy that is proposed to work, oftentimes does not always work. For example, here's kind of some of the latest checkpoint inhibitors or some HER2 positive therapies that aren't receiving as much success as hopeful. So these are some of the challenges that we hope to overcome by using custom models. Now, in doing so, by improving the translation using custom models, the ultimate goal is to get a greater success rate in FDA approval. But there's also some other endpoints that we can achieve by using custom models. This is to help accurately assess the mechanisms of actions, discover more biomarkers. We can help identify perhaps combinations of drugs that will work, or rethink how dosing schedules are performed. This also helps with stratifying patient populations. Again, the concept of heterogeneity. Therefore, the drugs that are being developed should be more targeted and focused on a smaller population to help improve outcomes of that specific group. In doing so, by creating better models, more translational models, we hope to reduce the false positives and false negatives that are seen in clinical research. And ultimately, this will help save time and money, reduce animal use. It takes anywhere from $1 billion to $4 to $5 billion to get a drug through the FDA. By improving the preclinical models, we can set that process up for better success and therefore reduce time, money, and animals. Historically, cancer research has relied on conventional models. Now, that's not to say that these models are not useful. These models have done extremely important work to help get us where we are now, but there are many limitations that have become apparent, especially in this day and age. Starting out with 2D models, traditional cell lines in a two-dimensional dish. Again, decades of research has been done on these, and they still have their place in biology today for understanding mechanism of action, understanding the biology. But as one would realize, that a two-dimensional model does not translate well to a three-dimensional disease. So therefore, there have been improvements, and I'll talk about the advancements in a little bit. Syngeneic models, on the other hand, is one of the latest type of models that are achieving a lot of attention due to the refocus on immunotherapies and checkpoint inhibitors. And the advantage of syngeneic models is that you have an animal with a complete immune system. However, the disadvantage is that it is a 100% mouse system. So the translation is lacking, which is pretty apparent there. Genetically engineered mouse models also, within that same light, tell the story and help identify the central biology of disease. But again, it is solely a mouse model, and thus does not have a great translation towards the clinic. To overcome this, patient-derived xenograft models have been used. That is by taking a human tissue, growing it in an immunocompromised mouse, and therefore the assays that are performed are actually performed on a human tumor sample. However, traditionally this is done in the subcutaneous fashion, where the tumor is placed under the skin. And as we know now, the tumor microenvironment plays a critical role in cancer biology. And therefore, subcutaneous tumors, while they are necessary and very important and have gotten us here, they still lack that translation. They still do not take the translation as far as it can, due to the discordance within the microenvironment. On the other hand, there's been a lot of advancements in advanced models. As I mentioned just previously, three-dimensional cell-based assays have become very popular and very widely used. Now, this could be simple from taking a two-dimensional cell line and then growing it in a certain matrix that makes it three-dimensional, which allows the investigation of penetrance of your cancer drug through that model. It can also involve taking a stem cell and growing it in a spheroid manner, where that stem cell divides and replicates in a three-dimensional fashion. And nowadays, one of the most exciting research being done is in organoids. These are multicellular, heterogeneous tumors grown in a dish in a three-dimensional manner that not only contain tumor cells, but also have the other microenvironmental cells, such as vasculature, immune cells, and other stromal components. With regards to the animal models, again, the conventional models have taken us thus far, but in order to achieve greater translation into the clinic, one of the steps that can be done is creating orthotopic models. This is by taking a human, in the case of PDX, taking a human tumor and growing it in the exact or the replicative organ of that the tumor came from. So for example, if a colorectal patient has a biopsy taken, this biopsy can then be implanted into the colon of the mouse, and therefore create a more clinically relevant model. And what is really exciting these days is the new generation of humanized models. So as we probably all know, immunotherapies is one of the greatest focuses in cancer research today, with over 60% of the new therapies in development in immunotherapies. Specifically in the case of checkpoint inhibitors, it's important that it's necessary that the animal model has a functional immune system. So by creating a humanized mouse model, this is where human immune cells are engrafted in the mouse alongside with the human tumor to be able to analyze checkpoint inhibitors and other modalities to harness the human immune system in a preclinical model. And finally, I'm just going to touch upon that there have been advancements in technologies, in equipment that can help model what a patient will go through. And specifically, I'm going to focus on a focal irradiation. So now there are irradiators out there that can do very guided focus beam irradiation. So you can take an animal model, treat it with standard of care chemotherapy, treat it with irradiation, and then from there, now you have a more relevant tumor that has seen a similar course that the patient has, and then you can assay your compounds on that, or in combination with irradiation. There are a number of ways to integrate these advanced technologies to help increase the success rate of drug development. So here's an overview of some of the latest models that I discussed. And it's important to understand that each type of model has its limitations and has its advantages. And really the best use of these models will be to identify the specific question that one is asking for. For example, if you have a new compound that's in development, and an investigator just wants to know what is the biological outcome. Can this compound hit the target and produce a biological response? Well, a 3D assay might be appropriate for that. If you have a new checkpoint inhibitor that is being development, a syngeneic model may be more appropriate for that one, just to see the biology. But ultimately, if you're at the end of this compound and are ready to go into the clinic, a more translational model might be appropriate for that. In this case, a humanized orthotopic PDX. Therefore, you are most replicating the human condition within the assay system that you're performing. So that is an overview of kind of the comparison of these models, and we're going to dive into some examples in more detail. But first, I think we have a poll question. So I'm going to hand it off to Aisha to display the poll question here.
That's right, Dr. Nakashima. So we do have a poll question coming up for our audience members to interact with in real time. And that poll question should be appearing on your screen. So the question that we have for you is, please specify your level of agreement with the following statement. Recapitulation of tumor microenvironment should be a requirement for preclinical models used in efficacy studies. And so the answer choices that are available are strongly agree, disagree, neither agree nor disagree, agree, or strongly agree. So once again, please specify your level of agreement with the statement that is displayed on your screen there. And the statement is recapitulation of tumor microenvironment should be a requirement for preclinical models used in efficacy studies. So please take a moment and select the appropriate answer choice that fits and suits you best. I'll give everybody a couple of more seconds to get their votes in, and then I'll close up the poll and share the results. All right, I can see that the votes are coming in. So I'll give everybody just another moment or two to submit their votes on this question before I close up the poll. All right, it looks like most of you have voted, so thank you very much for doing so. Now let's take a look at the results. So 14% of our audience responded strongly disagree, 0% said disagree, 10% voted neither agree or disagree, 33% came in with agree, and 43% responded strongly agree. So thank you very much, audience members, for your votes. Now it's back to you, Dr. Nakashima.
All right. So today I'd like to focus on a few examples of ways in which custom model development can aid in improving preclinical cancer research and discuss some of the opportunities available to utilize multiple approaches in order to generate the most clinically relevant data. I'll start off with utilizing cell-based models for in vitro studies, and then I'll move into some of the more advanced strategies for testing therapeutics in animal models. As Jonathan mentioned, one of the most important aspects of selecting a model system for preclinical testing is to understand the benefits and limitations of each particular system, as well as to understand how those assays selected translate clinically. As we all know, one of the most popular approaches early on in drug development is to start with an in vitro platform. In vitro assays can actually provide very valuable information with regard to things like target engagement, mechanism of action, and efficacy of a particular therapeutic agent. And of course, they could also provide a more high throughput and cost-effective platform for screening therapies prior to moving directly into a biological system or in vivo readout. Now, while two-dimensional in vitro testing has been in use for decades, it's become increasingly clear that three-dimensional in vitro systems, such as 3D spheroids, sometimes referred to as MCTSs, they offer an intermediate complexity between 2D and in vivo solid tumors. Most notably, in that they can help maintain the heterogeneity and expression profiles of the original tumor while also more closely mimicking the three-dimensionality or structure of the tissue from which they were derived. Now, before I jump into this particular model build case study, I'd like to mention that this was a highly collaborative effort with several institutions that we've worked closely with, including Tim Spicer and Louis Scampavia's group over at Scripps, Florida, Frank Feranis and Khalid Hanafi at Baptist Health, Samina Boca, as well as other contributors at Boca Raton Regional Hospital. And this study involves a patient diagnosed with glioblastoma, or GBM. For those of you who may not be as familiar with GBM, it's the most common form of malignant primary brain tumor in adults. And despite an aggressive treatment strategy, which includes maximal surgical resection, followed by concomitant chemotherapy and radiation, patients are left with a pretty grim prognosis as median survival times hover around 16 months. So needless to say, GBM is a devastating diagnosis for really any patient to receive. Now, in this particular case, the patient actually underwent a total of 3 surgical resections, all of which were able to be obtained and processed into patient-derived GBM cultures. The first resection, as is shown in the schematic, occurred in late 2018 to debulk the lesion. And at that time, the patient decided not to undergo any further treatment. A second resection occurred about 3 months later, after which the patient received standard of care treatment, which is, of course, temozolomide and radiation. And finally, a third biopsy was obtained after that treatment. Now, because we were able to obtain all 3 resections, we sought to understand just how much this patient's tumor changes over time. And to do this, with the help of our bioinformatics team, we performed a clonal phylogeny analysis using whole exome sequencing to identify the evolution of this tumor based on variant allele frequencies. And ultimately, what we found, as is demonstrated visually by the colors below the schematic, is that the tumor's genetic signature does in fact change over time, particularly following treatment. So when developing preclinical models, it is important to keep in mind that sometimes therapy is quite complex and can even be a bit of a moving target. Now, when looking at the genetic and molecular characteristics of this patient's tumor, it does have a methylated MGMT promoter, which traditionally indicates a better prognosis and response to temozolomide, standard of care chemotherapy. It also has a pathogenic BRCA2 mutation. And while there are drugs approved to target this mutation in cancers, such as breast and ovarian, they're not currently approved for GBM, and additionally, the particular variant expressed in this tumor is of unknown clinical significance. The tumor also expresses a missense mutation in TP53, which, as we all know, is one of the most commonly mutated tumor suppressors in a variety of cancers, including GBM. But drugs targeting downstream nodes, such as CDK4, CDK6, have proven largely ineffective in this particular cancer type. And then finally, the tumor has an amplification in EGFR, not uncommon in GBM, of which there are several classes of small molecule inhibitors available, but once again, drugs like lapatinib and erlotinib, which target EGFR, have failed to demonstrate efficacy in a clinical setting. So overall, the options for testing efficacy of a novel therapeutic using this information is a bit underwhelming. So instead, we utilized a different approach in which we combined, if I can get my slide to advance, in which we combined an in vitro drug sensitivity screening platform with drug gene network analysis. And once again, I would just like to acknowledge the work of Samina Boca, Tim Spicer, and Louis Scampavia in helping perform this assay. But ultimately, this analysis revealed 5 drugs with potential increased efficacy and synergy. I won't go too much into this slide for the sake of time, but upon testing those drugs in vitro in various combinations as shown here on the left, we were able to identify the combination of vemurafenib and cobimetinib in the upper right as a synergistic therapeutic option. And ultimately, this encouraged us to further develop these custom models in an in vivo setting. Now, before I move on, I would also just like to highlight additional opportunities for more in-depth testing that are available with regard to standard of care. In the case of GBM, standard of care involves temozolomide and irradiation, as I mentioned before. So depending on the therapy of interest, looking for potential synergy, or even a potential second-line therapy if there is a lack of efficacy in first line, we are able to ask more complex questions in an in vitro setting that more closely mimic real-life clinical strategies. These figures just show some of our GBMs irradiated at various doses with and without temozolomide treatment so that we can assess differences in sensitivity and synergy in vitro. And these types of studies can also be performed in an in vivo setting, as Jonathan mentioned, using our focal irradiator. Now, at this point, before moving into my next section involving in vivo model development, I'd just like to recap once again the importance of utilizing a variety of tools in order to ask and answer complex scientific questions. So in the study that we've been discussing thus far, for instance, we utilized expression analysis to identify clonal evolution over time to understand the heterogeneity and progression of the disease. We also used expression analysis to investigate the mutational profile that could be driving oncogenesis or resistance, where we saw MGMT promoter methylation, BRCA2 and P53 mutations, as well as EGFR amplification. We then utilized in vitro drug screens and gene drug network analysis to identify non-traditional therapies with the potential of synergy. And lastly, we've also discussed utilizing clinically relevant tools such as irradiation to help model the patient experience. So when working in the field of custom model development, which is the topic, of course, of today, it's important to recognize that different models, different assays can provide different pieces of a very complex puzzle. So with that said, let's move on to animal models for in vivo studies. Continuing on with the triple resection from our GBM patient, we decided to develop in vivo models by implanting all 3 cell lines as single cell suspensions using a side-by-side subcutaneous and orthotopic or intracranial implantation strategy. This was done with the help of the MRI in order to assess any differences in engraftment efficiency, while also allowing us to identify the growth kinetics of these models with which to establish a therapeutic window for future drug testing. Using this method, we were able to achieve a 100% engraftment rate in both locations, and we determined that these models do in fact offer a robust in vivo platform with which to test our in vitro screening results. I would also just like to bring up at this point that while the growth trends are similar, there are certainly differences in how these tumors engraft. Subcutaneous tumors engraft at a slightly accelerated rate and reach a larger size than orthotopic tumors due to a lack of intracranial growth constraints. And while not shown here in this particular slide, histological assessment postmortem did show noticeable differences in morphology and integration into the brain versus in the subcutaneous compartment. And of course, when considering a treatment strategy, one has to take into account the presence of the blood-brain barrier, which does not exist in a subcutaneous model. So as an initial proof of concept, we set up an in vivo pharmacology study for the GBM patient's initial resection, the BRRH001, which is the treatment naive. And we looked at varying doses of vemurafenib and cobimetinib alone as well as in combination, and ultimately found that the combination did prove to demonstrate synergy as shown in green with group 4. So overall, This particular case study represents just one example of how to effectively approach modeling cancer in a preclinical setting. Another aspect of in vitro custom model development to consider is with regard to metastasis. Now, metastatic disease, unfortunately, is the primary cause of death among cancer patients, and despite advancements in PDX techniques, translational models of metastasis are still lacking, and we realize that here at Certis. One example of how we can attempt to overcome this challenge is to develop bioluminescent patient-derived and PDX-derived cultures in order to utilize optical imaging to create robust translational in vivo models of metastasis. In this example, we developed a cell line from a lung adenocarcinoma that we then transduced with a viral vector to overexpress firefly luciferase. We then infused these cells systemically into animals via cardiac injection, and we utilized BLI to track where the tumor cells engrafted. We were able to identify signal in multiple areas within the animal, such as the bones, lungs, kidneys, liver, and other organs. And here are just some post-mortem representative histology and IHC images. The IHC here, we're looking at specifically human cells by staining for anti-human nucleoli. And they show morphological characteristics of this tumor infiltrating and engrafting in various organs throughout the animal. Here you can see on the left, engraftment in the lungs, bone, brain, liver, etc. And while we understand that this system is by no means a perfect model, this and similar experiments that we are currently performing do allow us to at least get a glimpse into some of the characteristics involved in late stage and spontaneous metastasis in an in vivo setting. The last example I would like to present is a pharmacology study in which we sought to design an assay to better investigate resistance and recurrence in an in vivo model of sarcoma. In this case, we had a recurrent myxofibrosarcoma originating in the skeletal muscle of the patient. I'll note that prior patient treatments are shown above the graph in the top left if interested. But we engrafted this tumor orthotopically into the skeletal muscle of the right thigh of animals and began treating with various drugs suggested by the patient's oncologist. So this was done in real time, side by side with the patient undergoing treatment. Fortunately, we were able to find that one of the 5 therapies, which was a combination of gemcitabine and docetaxel, demonstrated high efficacy. That's shown in the red line. And in that case, the tumors actually regressed down to being undetectable by our MRI imaging. And at this stage, it presented us with 2 opportunities. Number one, were we able to effectively cure these animals with this particular therapeutic regimen? Meaning, if we stop treatment, will it grow back? And number 2, if the tumor does grow back, is it now resistant to those therapies, or will it respond once again if we reintroduce them? So that is exactly how we proceeded. We stopped treatment and continued to monitor tumor growth by MRI. Now, between 3 and 4 weeks after stopping the treatment, we in fact did see recurrence. So we immediately placed the animals back on the gemcitabine and docetaxel regimen. This worked to inhibit tumor growth for a period of time. However, as is shown in this figure, the tumor eventually did stop responding to the therapy, and eventually the tumors took off exponentially despite the presence of the drugs, indicating that resistance was achieved. So just once again, depending on the question being asked, utilizing a custom developmental approach to model clinically significant outcomes can be of real value for a number of complex scientific questions. And with that, I believe we have another poll question, after which point we will be turning things over to Dr. Rajiv Sharmali.
That's right, Dr. Sperry. We have another poll question coming up for our audience members, and I'm going to launch that right now so that it should be appearing on your screen at the moment. So the question that we have for you here is, what types of clinical scenarios are you most interested in mimicking in your preclinical studies? Please select all the choices that apply. And the choices are prior treatment with standard of care drugs, radiotherapy, acquired resistance, metastases, or other. So once again, the question that we have here is, what types of clinical scenarios are you most interested in mimicking in your preclinical study? And you can select all of the answer choices that apply for your case. So I'm going to give our audience a couple of moments to submit their votes, and then I'll close the poll up and share the results momentarily. All right, I can see that the votes are coming in, so thank you very much for doing so. I will give our audience a couple of more seconds to get their votes in before I close up the poll. Okay, it looks like the majority of you have voted, so thank you very much for doing so. Now let's take a look at the results of the poll. So 33% responded prior treatment with standard of care drugs, 28% responded radiotherapy, 72% responded acquired resistance, 78% are interested in mimicking metastasis in their preclinical study, and 17% said other. So thank you very much once again to our audience for participating in this poll question. And now it's back to our speakers, back to Dr. Sharmali in this case.
Thank you, Jensen and Jonathan. We are glad to present our findings and perspective on PDX and humanized mice models for immuno-oncology application. Over the past decade, we have observed the field of immuno-oncology and cancer immunotherapy has grown exponentially with approval of immune checkpoint inhibitors, bispecifics, ADCs, cell and gene therapy, and other immune modulatory agents and their combination. Interestingly, especially for the large molecules like cell and gene therapy, the success has been limited to hematological cancers. This highlights the challenge and need of appropriate clinically relevant solid tumor models to test these drugs. As the field of immuno-oncology advances rapidly, there is a definite need of clinically relevant tumor models which can closely recapitulate the human tumor microenvironment and the patient population. At Certis, we believe humanized orthotopic PDX models are the holy grail and will provide us with the most clinically relevant tumor models to test various immunotherapies. To support this, I'm going to share some of our case studies to the audience. On this slide, we present the data for our lung adenocarcinoma model, which has been implanted subcutaneously in CD34 humanized NOG-EXL mouse. In the presence of a fully mature human immune system and functionally immunocompetent, you can see the combination of immune checkpoint inhibitors, Yervoy and Keytruda, drives a significant anti-tumor immune response. And when we look into the immune phenotyping of the tumor infiltrating lymphocyte, we can see an enhanced CD8, decreased T regulatory cells, which lead to an enhanced therapeutic index, that is CD8 to Treg ratio, in the combination group in the presence of human immune system. This tumor infiltration is in concordance with the IHC, as you can see. In the control group, it's populated with FOXP3 cells and very few CD8s, while in the combination, we see an increased concentration of CD8 T cells. We took this model to next level of challenge by implanting them by creating the orthotopic PDX avatars, where we implanted the luciferase tagged tumor line intracranially and also intrapulmonarily. And this has historically been thought to be a challenging model with low survivability, but at Certis, we have expertise in this domain. The mouse survives and we get very good implantation of this tumor topically and orthotopically. At the end of the experiment, since this was a NOG-EXL mice, we were interested to fully characterize the TILs and immunophenotype them. And you can see we are able to capture Various immune subtypes in the tumor include myeloid cells, their functional ability, B cells, T cells, and different subpopulations of T cells in orthotopically implanted tumor in CD34 NOG-EXL mice. So once we have this developed subcutaneous and orthotopic model, obviously the first question was, is there a difference in the immune response for a given tumor when implanted subcutaneously and orthotopically? In this colorectal cancer PDX model, we can see on the left-hand side, the immune response genes are one of the top regulated genes when the tumor is implanted subcutaneously or orthotopically. On the right-hand side, you can see the gene expressions analysis done, and very clearly, there is a subset of immune genes which are up or down, depending upon where the tumor is implanted. Our next question was, does the tumor implantation site also matters for T cell recruitment to the tumor site? In this recently concluded study at Certis, we have used this gastric model where the tumor was either implanted subcutaneously or orthotopically. And on the left-hand side, you can see in both the cases, the immunization window was between 2 to 3 weeks, where we can see the required number of CD45 in circulation. And over a period of time, 2 to 3 weeks further, we see a very nice, kinetics of CD45 in periphery, but there was no difference in the rate of immunization. It was similar between subcute and orthotopic percent CD45 in periphery. We terminated the mice, and on the right side, you can see the immune phenotyping of different T cells. In blood, on the top right, we see CD45, CD3, CD4, CD8. There is no difference in the periphery. Interestingly or intriguingly, if you look at the PDX at the bottom, there is a distinct difference in the T cell recruitment depending upon where the tumor is implanted. The subcutaneously located tumor has more infiltration of T cells, while the orthotopically located tumor has reduced number of T cells, CD45, CD3, CD4, CD8. We are testing this observation across different donors and across histologies. Given this initial observation where we see or we have evidence that the tumor implantation site matters and there's a differential immune gene signature and preferential recruitment of T cells depending upon the tumor implantation site, obviously the next question was, does those observation drives a difference in outcome of immune response efficacy? In this liposarcoma model, we have a subcutaneously implanted tumor in the skin and orthotopically where the tumor was implanted in the bone. And we tested the effect of anti-PD-1 treatment on the same tumor when it's implanted at 2 different sites. And the mice were immunized with PBMCs. You can see in the subcutaneous model, the anti-PD-1 drives a 30% TGI by the end of experiment. While in orthotopic model, the same PD-1 failed to drive any efficacy. And when we looked into the tumor infiltrating immune phenotype at the end of the experiment, it was interesting to observe that in subcutaneous tumor, the CD4s had lower PD-1 or low exhaustion and had CD8 with higher effector function ability of interferon gamma and gray enzyme secretion. While in the subcutaneous tumor, while in the orthotopic tumor, there was more PD-1 on CD4s infiltrating the tumor. And so that means there is more exhaustion of the CD4. And the CD8s were less functional or has less effector ability of secreting interferon gamma and gray enzyme compared to the subcutaneously implanted tumor. So from our preliminary observations, when we compare the implantation site, subcute versus ortho and effect of immunotherapy, it definitely exhibits that there is a differential immune response and it may determine the outcome, including immune gene signatures, T cell recruitment to the tumor site, and also the efficacy outcome of any immunotherapy, especially immune checkpoint inhibitors. So we wonder, these initial observations, what we have, is is some of the mechanism of action for the disconnect we see in the preclinical testing and the clinical observations for various immunotherapies. Thank you.
Thank you very much, Dr. Shrimali. And now we have a couple of poll questions to conclude the presentation before we head into the Q&A session. So audience members, your participation once again is greatly appreciated. So let's take a look at one of the two poll questions that we have for you here. And this question is asking, when testing immuno-oncology therapeutics in vivo in humanized PDX models, what is your preferred implantation site? And the answer choices available are subcutaneous, orthotopic, including primary and any metastasis sites, ectopic, other than primary or metastasis site, or no preference, or perhaps other. Once again, that question is, when testing immuno-oncology therapeutics in vivo in humanized PDX models, what is your preferred implantation site? So please take a moment and select one of the options that best suits you. I'm going to give our audience a couple of moments to get their votes in before I close up the poll and share the results. All right, I can see that most of you have voted, so thank you very much for doing so. I'm going to close up the poll and share the results here. Okay, so we have 0% for subcutaneous, 79% for orthotopic, 14% for ectopic, 7% said that they have no preference, and no votes for other. So thank you very much once again for your participation in that poll question. And we have one final poll question to round out the presentation, and that should be appearing on your screen. So once again, we really greatly appreciate your participation. And this question is pertaining to PDX models. And the question is, when selecting PDX models, what characterization data is most important to you? And for this question, you may select all of the answer choices that may apply. And the choices available are growth curves, patient treatment history, in vivo pharmacology data, gene expression or whole exome sequencing results, or other. And once again, that question is, when selecting PDX models, what characterization data is most important to you? And you may select all that apply. So once again, we would greatly appreciate your participation, and I'm going to give our audience just a couple of more seconds to get their votes in before I close up the poll here. Okay, I can see the votes are coming in. Thank you very much for that. I'm just going to give you another moment or so to get your votes in. And I think that does it. So let's close up the poll and look at the results. All right, 25% responded that growth curves are an important characterization data when selecting PDX models. And 25% said patient treatment history, and it's split evenly across the four answer choices actually. And then 25% also for in vivo pharmacology data, and 25% for gene expression or whole exome sequencing results, and no votes for other. So once again, thank you very much to our audience for participating in this poll question and all of the other poll questions as well. We greatly appreciate your participation. And now it's back to Dr. Nakashima, who will round out the presentation before we head into the Q&A.
Thank you, Aisha, and thank you everyone for taking those poll questions. So just to wrapping up just a little bit about us. Certis is a translational science company. What we do have online is our cancer model database. This is called Barney OI. It is named after our chairman's late son, who unfortunately passed due to cancer. But along his journey, we at Certis did create a model for him. and was able to use that model really in research. So you can log on. Again, at the end of the presentation, there'll be some contact information. But our online database, Barney has all of our PDX models on there, about 180 right now, and growing. We have another 200 plus or minus in development. And with those, you can, it's often like a Google search, so you can drill down and find the genetics of these models, patient history, as well as model characteristics such as growth curves, pharmacology, and what have you. Again, we are Certis Oncology Solutions. We're located in San Diego, California. We are CLIA certified, and we use orthotopic PDX models to help identify therapies for a patient in real time. We use these models to also advance drug development and to help provide what our product is, which is Oncology Intelligence, a end-to-end whole information regarding these cancer models and the response due to your therapies. So with that said, I'm going to end here. Again, you can get in touch with us. The contact information is here on our left, as well as additional resources on our webpage to learn more about us and the services that we can provide you. And with that, we'll conclude the presentation and open it up to questions.
That's great. Now, thank you very much to our speakers for that very insightful presentation. And we are now heading into our Q&A session. So please feel free to keep sending in your questions and comments for our speakers, and we will try to attend to them in the Q&A session. So I've already received some questions, and we can begin with those. So the first question that we have here is, apart from treatment, for example, in the GBM case with TMZ and radiotherapy, are there any other factors which could change the tumor genetic composition?
Okay. Take that. Sorry, once again, apart from the treatment, are there any other factors that could change the tumor composition, the genetics? Well, I think, one second. What we do, what we've observed and kind of demonstrated in the gene expression is that the microenvironment can play a role in the gene expression. Now, that's not to say that it'll change the actual physical mutations within the DNA. That we haven't observed yet, but that is also a possibility. Again, when you create models, every time that we grow something, we're selecting for. So we're well aware that as we passage these models, as we implant it orthotopically, ectopically, sub-Q, there is a selection process that undergoes. In our analysis, that selection process hasn't resulted in anything profound changes, but we can't rule out the possibility of that. Pass to my colleagues if they have any other comments regarding that. So I think we're good. And again, we had a little technical difficulties with our audio, so I apologize if there's some background there. We're trying to work that out right now.
Everything sounds great, so nothing to worry about. All right, so let's move on to another question. And this question is, aside from differences in tumor growth kinetics, what other differences have you observed between orthotopic implants versus subcutaneous implants?
Great question. Jonathan had to mute, sorry. So we've actually observed several differences. We've observed differences in expression. We actually presented a poster on this at AACR. So there are significant changes in expression between orthotopic and subcutaneous. Morphology, as far as how the cells integrate within certain tissues. Certainly in the case of the GBM, certain GBMs will form a nice tight ball, whereas others are a little bit more diffuse with regard to similarities to a diffuse astrocytoma. We've seen differences in drug sensitivity. Once again, since we've talked about the GBM in this case, you have to deal with a blood-brain barrier in the case of if you treat orthotopically versus subcutaneous. some differences in vascularity, and then also what Rajiv touched on too is just with regard to T-cell infiltration. So just depending on the site that you engraft, you can actually get a different immune response in the orthotopic versus subcutaneous environment.
Wonderful. Thank you very much for that answer. Another question here is, what are the current limitations or difficulties for using orthotopic PDX models in preclinical research?
Sorry, just gonna have to button there. The limitations in orthotopic. Well, one, it's more challenging. It requires surgery, whereas subcutaneous, again, there's different modes of subcutaneous. You could do an injection with ECM, such as matrigel of single cells. That's probably the easiest. Oftentimes, you don't even need to anesthetize the mice. What we do here at Certis is that when we do subcutaneous, we do tissue fragments. So that does require some minor surgeries, anesthesia, suturing, or staples. With orthotopic, depending on the site, if you're doing a thigh versus intracranial, there's obviously a wide range of complexity within that. So for advanced models such as intracranial, you have that limitation, meaning it takes more time to implant. What we do here at Certis too is that you have to make sure that the cells actually deposit in the brain and are not retrieved when you retract the needle. So that just adds more time to that. It also requires special surgery. We have stereotactic instruments to precisely inject tumor cells within the brain. For other types of other modalities, such as the bone or the lung, that also requires injections versus surgical tumor fragments. So that's probably one of the main differences is one, the complexity of creating the model, and the second one is monitoring that model. Subcutaneous tumors can be monitored by using calipers, whereas orthotopic models need some non-invasive imaging. So we overcome that by using MRI. We also, as we demonstrated, we have luminescence. But again, there's a little added to that. In order to use bioluminescence, you have to create a model that's expressing luciferase. So that's one more process in developing a luciferase based model. So with all that taken account, orthotopic is more challenging technically from creating the model as well as monitoring the model as well. But we've invested in the tools and the capabilities to overcome those limitations.
Wonderful. Well, that brings us to the end of the Q&A session and the end of the webinar today. If we couldn't attend to your questions, the team at Certis Oncology may follow up with you, or if you have any further questions, you can direct them to the contact information, the email address displayed on your screen there. I'd like to thank everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen, and your participation is appreciated as it will help us to improve on our webinars. Now, I'm about to send you a link in your chat box, so let me just do that right now. And you'll be able to view the recording of this event on this page. And you can also share this link with your colleagues when they register for the recording here as well. So I do encourage you to do so. Now, please join us in thanking our speakers. Today, Dr. Jonathan Nakashima, Dr. Jansen Sperry, and Dr. Rajiv Shrimali. We hope you found this webinar informative. On behalf of the team here at XTalks, thank you for joining us. Please take care and bye for now.
Thank you, everyone.